Hello everyone, so today I will be telling you about structure, life cycle and pathogenicity of Wuchereria bancrofti. Wuchereria bancrofti, first of all, scientific classification. It belongs to kingdom Animalia, phylum Nematoda, class Phasmida, order Filarial, family Filariidae, genus Wuchereria. Wuchereria bancrofti is commonly known as filarial worm. This worm causes filariasis in humans. So what do you mean by filarial worm? Filarial worm is something which appears like thread. So basically the body or the shape of the Wuchereria bancrofti is thread-like. This worm causes filariasis in human. Filariasis is a disease commonly known as elephantiasis. In Hindi we say it hathi pav, and it is a rare condition in humans. Demarque first discovered the larva form of this worm. So firstly, the scientist who first discovered is Demarque, and he discovered the larval stage of this worm. Later, Wucherer found these larvae in the Calaris urine. Later, Wucherer found the larva in the Calaris urine. Calaris urine is a rare condition in which the urine appears milky white. Lewis found them in human blood, and Bancroft found in the adult females. The name of these worm is given Wuchereria bancrofti in honor of the two scientists Wucherer and Bancroft, who made considerable contribution in studying the disease caused by these worms. So basically the Wuchereria bancrofti is name given in the honor of two scientists, that is Wucherer, who found the larva in the Calaris urine, and the Bancroft, who found the adult females. Distribution. This pseudocoelomate roundworm is spread throughout the world except in the polar region. It is prevalent in India, Arabia, Malaya, Korea, Japan, Brazil and South Pacific Iceland. It is relatively absent in Europe, North America and Africa. So basically it is a cosmopolitan except from the polar region and Europe, North America and Africa. Habit and habitat of Wuchereria bancrofti. This filarial worm is dreadful human parasite residing in the human bloods and lymph. It is digenetic parasite requiring two hosts, digenetic, and it completes its life cycle in human and in the Culex mosquito. The adult worms are harbored in coiled state in human lymph glands and lymph passages. These worms obstruct the passage of the lymph. Now, the structure of filarial worm. The body of adult filarial worm is elongated, narrow, filiform and cylindrical in shape. These worms are creamy white in color. Anterior ends terminate bluntly, whereas the posterior end is little pointed. The male and the female worms are found coiled round each other in the lymph vessels. A mouth with no oral lips is present at anterior end. The alimentary canal of this worm include mouth, pharynx, esophagus, intestine and the anus. The anterior part of the pharynx is muscular and the posterior part is glandular. So the anterior part of the pharynx is muscular and it is glandular in shape. Wuchereria bancrofti is unisexual in nature, exhibiting sexual dimorphism. So basically the female and the male are different. The female worms are slightly longer. As you can see that the female worms appears to be long and it is thicker than the male. The female worms are slightly longer and thicker than that of their male counterpart. Each female worms measure around 65 to 100 mm in length and 0.25 meter in diameter. So it is around 65 to 100 millimeter in length, and in diameter it is around 0.25 millimeter in diameter. The posterior end of the female is straight and bears anus. The posterior end is straight and bears the anus. The female genital pore, also called as vulva, is present ventrally. So here the female genital part is also present, which is known as vulva, and it is present ventrally at about 1/3 length of the body from anterior tip. So from the vulva is present from the 1/3 of the body, from the anterior part. Male worms are comparatively smaller and thinner, as you can see in the picture also. The male is thinner and it is smaller in shape than the female one. And each male filarial worm measures around 40 millimeter. It is around 40 mm in length and 0.1 mm in diameter. The posterior end of the male worms is curved, bearing cloaca. So you can see that the anus part is straight, the posterior part is the straight, and but in the male it has a anal style and it is somewhat round, or you can say curved, and it bears cloaca. In the region, a pair of unequal penile setae or copulatory spicule is present in cloaca or curved region. Many copulatory papilla are present in the posterior region. So many papillae is present in the posterior region, right? Copulatory spicules is present in the posterior region, and it is somewhat is curved, right? It is somewhat curved than the female one, and it is known as anal style. Now we will be seeing about the life cycle of Wuchereria bancrofti. So I have told you that Wuchereria bancrofti is digenetic in nature, means it requires two hosts for completing its life cycle. The first host is the human and the second host is the female Culex mosquito. So here we will be discussing first is the life cycle in human. This cycle is also known as human phase. So the cycle is also known as the human phase. The process of copulation takes place in the lymphatic system of the human, and the male and the female is unisexual in nature. So here the copulation takes place in the lymphatic system of the human. The male and female worms copulate when they are present in the same lymph gland. The female worms are ovoviviparous. The female are ovoviviparous means they produce eggs which hatches within the body of the female without obtaining nourishment from it. So as usually in oviparous or in viviparous, in oviparous they lay egg, but in viviparous the placenta is present and they give birth to the young ones. But in ovoviviparous they produce egg and the egg hatches within the body, right, without taking the external nourishment. Numerous that is called placenta in viviparous, right? Placenta is absent, basically absent in the ovoviviparous, and they obtain the nourishment from the eggs, the material of the egg. Numerous microscopic juvenile larvae called microfilariae are released into the lymph. So as a result of copulation, numerous microfilaria are released into the limb. Each microfilaria is about 0.2 to 0.3 mm in length. It is surrounded by a loose cuticle sheet. The cuticular sheet is also known as egg membrane. The surface of larva is covered by flattened epidermal cell. So the larval surface is covered by flattened epidermal cells. Its body contains columns of cytoplasm with a number of nuclei. An oral style is present at anterior end, whereas the mouth develops in the future. Right? So first of all, in the larval stage, they are covered, the surface is covered by epidermal cells, that is flattened epidermal cells, and the anterior style is present, and mouth develops in the future. So here the mouth is absent, right? And they contain cytoplasm and nuclei in the columns. So here you can see the diagram of Wuchereria microfilaria. So here the cuticle body sheet is present, oral style is present at the anterior part, nerve rings are present, bladder is developed, and nephridiophore is also developed. Anus, 4 large cells are also present in the microfilaria. Rennet cells, epidermis, and somatic cells are also present. You can also see the interior mass. So here a nerve ring around the pharynx, as you have seen, this is a pharynx, and the nerve rings around the pharynx is present. Exterior pore is present, that is the anus. Rennet cells is also present. 4 large germinal cells, future anal pore, inner cell mass, somatic cell mass is also present in the microfilaria. So the microfilaria, after being released into the lymph vessel, enters the blood vessels and circulate within the blood. Ultimately, they migrate to the visceral organ and resides in the deeper blood vessels of the thorax region. In the thorax region, the larvae do not undergo any additional change, as further development can happen only inside the intermediate host, that is mosquito. So basically, after the releasing of the microfilaria, they resides in the deeper blood vessels of the human, and the further development does not change, as the development will occur in the Culex mosquito. So in the daytime they resides in the deeper blood vessels, but in the night time they appears on the surface of the periphery of blood vessels, right, which suits the feeding habit of the mosquito. So these larvae moves to the periphery blood vessels during the night time between 10 p.m. to 4 a.m. to suit the feeding habit of mosquito. This movement of larvae is called as nocturnal periodicity, and this is known as the nocturnal periodicity. During the daytime, these larvae lives in the large deep sheet blood vessels, but during the night they migrate to the peripheral blood vessels, and this process is known as nocturnal periodicity. Hence, it can be said that microfilaria show day and night periodicity, also known as diurnal rhythm. The microfilaria die if they are not transferred to the mosquito within 70 days. So you can see that period is very long, that is 70 days. The microfilariae is the infective stage of the mosquito, and the microfilariae is the only stage, infective stage of the mosquitoes, right? Now next we will be seeing the life cycle in mosquito. This cycle is also known as mosquito phase. In human it is known as human phase. In mosquito it is known as mosquito phase. When the female Culex mosquito sucks the blood from the Wuchereria infected person, the microfilariae from the peripheral blood enters the midgut of the mosquito. So earlier I have told that the microfilariae in the night, which suits the feeding habit of mosquito, they appears on the peripheral blood of vessels in the human, from where the mosquito sucks the blood from the infected person, and the microfilariae enters the midgut of the mosquito, and then they shed their protective sheet within 6 hours. The protective sheet around the microfilariae is shed within the 6 hours in the midgut of mosquito. After shedding their sheet, they penetrate the stomach wall and migrate to the thoracic muscles, where they metamorphose and grow. So in human phase there is no further development after the microfilariae is formed, but in mosquito most of the development of the worm occurs, and they metamorphose and grow. Initially they metamorphose into flat sausage shaped larva, also called as first stage larva. So the first stage larva, after metamorphosis is formed, later this larva undergo first molting and grows into slender elongated second stage larva. Finally, this second stage larva undergoes second molting, transforming into a long infective stage, also called as third stage larva. So first of all, it is shed after entering the midgut of mosquito. They shed their protective layer within 6 hours. When the protective layer is shed off, they enters in the stomach wall and migrate to the thoracic muscles. Now here the first metamorphosis occur, and after metamorphosis first molting occurs. Then the second stage larva undergoes second molting and transforming into a long infective stage, also called as third stage larva. All these changes takes place within 10 to 20 days. So it requires around 10 to 20 days. The final third stage larva moves to the labium of the mosquito so that it can be transferred to the definitive host man. So basically, in earlier the infective stage is of 70 days in mosquito, but here the changes takes place within 10 to 20 days, and finally the worms, the larva moves to the labium of the mosquito so that it can be transferred to the definitive host man. Means they can transfer the infective stage of the larva into the healthy man, right? In man, the filarial larva first enters the blood circulation through the bite of mosquito, and then it enters into the lymphatic vessels, where it undergoes third and fourth molting and transforms into adult. The adult male and female worms copulate, and the female delivers the microfilariae, and again the cycles completing, right? Now, pathogenicity of Wuchereria bancrofti. Usually, it is transmitted from mosquito biting, but it can also be transmitted when the blood transfusion occurs from the donor, which has microfilariae in the lymphatic blood vessels, right? Filariasis is usually transmitted to man through mosquito biting. The disease can be accidentally transmitted through blood transfusion when the donor is infected with microfilariae. Filariasis or elephantiasis is the disease caused by Wuchereria bancrofti in human. These filarial worm resides in the lymphatic vessels of man and obstruct the flow of lymph, thereby causing severe condition known as elephantiasis, in which the limbs and other body parts grow into enormous size. So basically, the microfilariae which resides in the lymphatic vessels cause the lymphatic vessels to swell up, and the infected body parts appears to be enormous in size, right? They grow into enormous size. Now, pathogenesis. Light infection causes filarial fever. So in the earlier, you can say in the earlier stage of this worm or of this pathogenesis, the filarial fever, light fever, mental depression and headache also caused in the earlier stage of the infection. The infection of the parasite causes inflammatory effects. The inflammation in the lymph vessel is called as lymphangitis, and the inflammation in the lymphatic gland is known as lymphadenitis, right? Heavy infection, in light infection it causes fever, mental depression and headache, but in heavy infection, when the microfilariae density is more than 20,000 per milliliter of blood, in this case the accumulation of dead worms and the inflammatory reaction of the worm blocks the lymph vessels and glands, resulting in immense swelling in the extremities of the limbs. Scrotum of males and mammary gland of females. The condition of the swelling of the extremities is called as lymphedema, right? The swelling is known as lymphedema. The increased permeability of the wall in the lymph vessels lead to accumulation of lymph in the tissue. The fibroblast accumulate in the edematous tissue, causing fibrosis. This fibrosis tissue is formed in these region. So here, when heavy infection occurs, most of the around 20,000 per milliliter of blood, the lymph vessels are blocked because the number is very large amount, right? And the condition of the swelling of the infected area also occurs, right? In severe cases, the sweat gland in affected region disintegrate, causing the skin to become dry and rough. Other manifestation of this disease include lymphangiovarics, dilation of lymph vessels in inguinal, scrotal and abdominal region, lymphorages, rupture of lymph vessels, hydrocele, accumulation of fluid due to obstruction of spermatic cord, occult filariasis, hypersensitivity reaction to filarial antigens. Diagnosis includes the study of microfilariae. After staining, microfilariae of different species are identified based on their specific shape and morphological characters. Therapy and control includes complete satisfactory treatment, it is not yet known though the disease can be reduced or eliminated by eradication of microfilariae from the circulation by administration of hydrazine and compound of antimony and arsenic. Prevention: filariasis can be controlled by the following methods. Protection against adult mosquitoes. The adult mosquito can be controlled by spraying the insecticide like organochloride like DDT, BHC in the homes. Fumigation of dwelling places is also effective measures to kill the mosquito. Using mosquito net is the safest method to avoid the mosquito bite. So basically prevention is better than cure. So as a prevention you can use DDT, BHC, spraying of insecticides, right? Or you can usually use mosquito net, which is the safest method to avoid mosquito bite. Destruction of mosquito larvae: spraying of kerosene pyrethrum oil in the sewage, gutters and ditches is recommended to kill the mosquito larvae. Insecticides are used in breeding places to kill the larvae. Biological control using larvivorous fishes like Gambusia is much safer, right? Gambusia eats the larval stage of the mosquito, right? So it is a biological control, you can say. Thank you so much for the patience hearing. Bye.